So this might be too large of a group to do introductions for everyone, but I thought maybe it would be helpful if the folks at the Allen introduce themselves. And then that would give more time for people to come in. So typically we for every OpenScope project we do a kickoff where when we start the data collections, we get everyone in the same room and discuss the plan for the year. This project is very different, and it's the first time we're doing this. So this will be the equivalent kickoff. Given that the group is very large, going through everyone would take too much time. But I think it will still be helpful if the people at the Allen introduce themselves, so that you know who is working very hard on our hands. So maybe I'm going to sort of point at people that are at the Allen Institute so for them to introduce themselves. So maybe Ivana, you want to start?
Hi, I'm Ivana. Sorry, Hannah and I are in the same room. I'm the project manager of OpenScope. So with Hannah, we work on coordinating the project, communications, notes in the meetings, yeah, and all that. Welcome,
Anna?
everybody.
Anna?
Yeah, my name is Hannah. I am a project coordinator at the Allen Institute. I work very closely with Ivana and Jerome and the experimental teams to make sure everything is going smoothly and we get all of the data done.
All right. What else do we have from the Allen? Maybe Casper.
Hi, everyone. So I'm Casper. I run an experimental team here at the Allen Institute, and we have some sort of some unique imaging technology, especially for imaging dendrites, so glutamate and voltage in dendrites and things like that. And we are interested in doing some of these experiments.
Severin?
Hi, I'm Severin. So my team, which is Ryan and Mickey, are collecting the electrophysiology data for OpenScope for a few years now.
And Jimin?
Hi, I'm Jimin. I'm a research associate at the Allen. I collect 2-photon data for OpenScope projects.
Okay. I saw David as at the Allen, but maybe you want to briefly introduce yourself?
Yeah. Hey, I'm David. I'm also working at the Allen. Not technically part of the OpenScope team, but I was on an OpenScope project a few years ago. And yeah, just interested to see how this experimental phase of this project is shaping up. So
we have Peter. If you can speak, I don't know. Yes.
Hello, everyone. Hi, I'm Pete Grobleski. I'm the Director of Research Operations. I've been working with Jerome on OpenScope for 5 years now, Jerome, maybe 6 years.
Yeah, fair amount.
I oversee the surgery, behavior, and optical physiology teams who help to collect the data. So welcome.
Carter? Can you hear us, Carter? Oh, you're muted. Oh, Carter had a lot of laptop issues in the last few days. So Carter has been building the data book that some of you have used, the OpenScope data book, and he's a data analyst on the OpenScope team, and I'm so thankful for him to be working here. I think we have introduced all the Allen folks. So why don't we get... Oh, Sheila is here. Sheila, are you able to introduce yourself? Oh, she disappeared? No.
Hi there. Hi there, I'm Sheila. I'm an RA on the planar OPHYS optical physiology group. We also do intrinsic signal imaging. Nice to meet you all.
Okay. So as I said, we just go through the Allen folks because there's a large number of people on the call, and so that would be more efficient. But so let's get started. So the agenda of the meeting is essentially we felt that going through our resources would be helpful. And as I said, this is the first time we are doing a public kickoff, and so we're gonna learn as we go over the years as we collect this data. So I hope you're tolerate some mistakes on the way, so that we will do our best to correct any errors as we disseminate this data to the public. So the first thing I want to do today is work through the project website that we are building to support disseminations. And then we'll briefly summarize the project goals. So those were extracted from the archive publication that we put together. And then we want to spend a little bit of time going through some collaboration guidelines. And our goal here is to potentially answer any questions or receive any feedback you may have in the group about those guidelines. Our intent is to sort of set the expectations clearly from the start. We've done a lot of collaboration over the years, and this is important. Then we'll discuss how we're going to track progress on our hand, and perhaps discuss how other teams that want to collect data will do so. And then we'll discuss a little bit about the stimulus design and deployment that we have been working on. And our intent there is to align on timelines. And then if there is not enough times, we should discuss analysis plan. And what I particularly care about is that we leave the meeting with a set of clear groups so that we can focus our efforts in the coming weeks. Are there any questions on this agenda that we should address before starting in the group? Okay, let's get started. So this website is a GitHub repository. So the reason we did this is to allow anyone to make edits. So it's a public repository that anybody can contribute, and we intend to build it as the project unfold to document the project, the dataset, and everything around it, as well as project management. So it's meant to be a single place where anybody can get informations about the project. Because obviously onboarding people is going to need good documentations. So it has a few important sections. The first is the project plan. So that's derived from the archive review and experimental plan that we put together with the community. It has project resources. So those are highly documented pages that explain the stimulus code, how to deploy it, the hardware, and then experiments that are ongoing, so that if you're interested in doing analysis on this data, you know what's going on and how to access this data set. And the last section is about project management. So this is to be a public face to highlight who is working on that, what is the way we work together, and then where we are on individual sub-projects. So I think it's pretty self-explanatory, but I'm going to go through these sections in the presentation today. So the project plan is highlighted in the overview here, and if you want to learn a little more about it, you can go through the archive paper that is now published. But essentially, we made a review of the predictive processing literature, and eventually design a set of experiments that are meant to be conducted after this review. These experiments are described here. And so I think what we found in the review is that there are a number of designs that have been used in the community to test predictive processing ideas. And there are also a number of modeling papers that have been pushed. There actually is weak overlap between those two communities, I would say, that's fair to say. And we think that by creating a bridge, so a data set that connect multiple experimental design in the community and make this really public, we can facilitate this iterative community building effort. That's the first thing. The second is among all of those papers that we review, we saw that the different experimental designs are highlighting different computational primitives. So, for instance, in the standard motor mismatch literature, so this is when you're presenting a sensory and a motor inputs to the cortex, and then it tries to predict one from the other. We found that the community has highlighted certain pathways that are involved and in between neurons. And that result is perhaps different if you look at standard mismatch experiments, where now to make the predictions, you use a very recent history of a visual stimulus. And so the review highlighted, but it's likely that depending on the sensory context and the prediction context, you might recruit different mechanisms to make predictions about the future. And so the goal of this set of experiment is to test this hypothesis that there are multiple mechanisms at play. And the intent is to help consolidate the community around a set of shared data set that can link with individual lab work. So that's the central idea of this set of experiments. And we highlighted the need for multiple modality data, so 2-photon data, as well as neuropixel data, as well as potentially dendritic imaging. And thanks to the NIH support that has continued so far, we will execute these experiments this year. Some of these under OpenScope support, and some of us through collaborating labs that are contributing experimental bandwidth for this effort. And so those are currently Sarah Rudiger, that is here, Kasper Podgorski. I think Karim OS also is interested in contributing, Andrea Bastos and Alex Meyer, as well as Farzaneh Najafi. I hope I don't forget any. And so those individual labs are interested in collecting data on one or several of these modalities. So Andrea Bastos and Alex Meyer are interested in collecting in complementary to mice primate data with this different session oddballs. Kasper Pogowski labs and scientists in his team are interested in dendritic imaging, but is highly relevant to many models of predictive processing. Farzaneh Najafi is interested in the duration mismatch session types, as well as Sarah Rudiger. And so they likely could collect 2-photon or neuropixel data respectively in this design. Did I forget? And then Karim always is interested in the sensory motor mismatch paradigm. So as you can see, there is a potential for multiple complementary data set that will be collected as part of this effort. And our intent is to make those data set immediately available to the community at large, so that the analysis plan that is described in the archive paper can be executed in a distributed fashion. And so the goal of this website is to help coordinate the data collection of this project, as well as the analysis part. So that's our goal. Are there any questions on the experimental goals of the project that we should answer here?
Are the experimental designs fully solidified? Like, is there no room to further contribute to experimental design?
Oh, that's right. Good question. So I think the experimental design that is what we plan to run as part of OpenScope, the NeuroPixel and the 2-photon, I think is fairly well defined at this point. The way I see it is all of this auxiliary, or let's not say auxiliary, but satellites data set, for instance, one in your group, I think are more free. I think it's important that they have point of contact with the main one we collect, but I think that's the intent. I also want to recognize that it's going to be challenging to have integrated homogeneous data collection environment across all of those labs. And so I don't think the spirit is to force that, because I think the field has tried this before and it's very challenging. But we will try to share. I would like to try to share the stimulus script at the base. So that's why we're going to talk a little bit about Bonsai later on.
Yeah, I'm just imagining a situation where as we start collecting data, we sort of run into unforeseen sort of things that are either very interesting and cool, or that, that don't happen, that, a lot of that we were expecting to happen and things like that. And if we wanted to sort of iterate, essentially.
That's right.
But that's for the future. Thanks a lot. That was a very clear answer.
I do want to mention that the stimulus parameters that are chosen here in this 4 session types derive from already established session types that have been published. So the majority of those parameters have been shown to work. And so we don't, for OpenScope dataset, we don't intend to explore there. We're just going to replicate as much as we can experiment design that were already published. Okay. If there are no more questions, I'm going to move on to the collaboration guidelines. I think that's related to Casper's question. So in the project management page here, you see a number of subpages, and one of these is a team page. So these people listed here are all the ones that contributed to the archive piece. And I want to emphasize that this is anybody can be added to this list. This is really open. We don't intend to enforce anything here except openness and for the list present here. The reason is I personally want to make easy for people to contact each other, and so having this make it really visible. But the thing that we do care about is that because as this project unfolds and people make discoveries and get excited about the data set or dive further, it's important that we agree on how we intend to collaborate. And so that's why we put together a number of things. So the first thing is the collaboration policy. And is there a question? Oh, yeah. Can you see the collaboration policy?
No.
Oh, you can't see that? What is going on?
Jerome, you might need to unshare and then reshare your other screen. Sometimes that helps.
Lost Jerome altogether.
There he is.
Sorry, it looks like my Teams was somehow frozen. Does that work now?
Yep. Yes, looks good.
Okay. So this document was we received some inputs from a couple people in the group here, and I think the intent is to sort of write down very simply how we intend to collaborate. And the first thing is we highlight that we want to sort of follow a very open and respectful approach for collaboration, where IDs, data, and code flows really freely. But it's very important that we give credit when credit is due, and that's for so that everybody feel like they are empowered, but also are rewarded for the work that they do. So to facilitate this, we think we should follow a very open model where data is open and unrestricted and flow. But to assign credits and make sure that everybody is properly credited, we want to establish an opt-in process where people that are interested in future manuscripts and feel like they should be listed as co-authors can say so. And so the intent is in the future, if there are derived publication from this work, we expect people to sort of publicly share ongoing manuscript and respectfully ask people to sort of that are part of this group and listed in the team page whether they feel like they should be co-authors. So that's the first thing. Essentially the same thing we expect to happen with the code, so very open sharing. And for project tracking, we will use the project tracking page that I'll discuss later on, that will list all of the ongoing projects. So this is the reason why this website is very easy to edit. So if you go at the top right of the page, there's the edit button. Anybody can go there and make changes. The intent is that whenever someone starts on a project, you can list it in the project tracking page so that people can join or are aware that you're working on a particular piece. And eventually, we think it's important that people that contribute dataset to this effort are given the opportunity to opt in, especially in the early times. So I think we listed guidelines at the top where is it listed? That's right. So given that there are the Allen Institute and also other contributing labs, we think in the first two studies that come out of a data asset, a grad student or postdoc or folks at the Allen Institute should be given an opportunity to be co-authors on the first two papers. If there are conflicts, we hope to put together a small steering committee composed of the people that are contributing dataset and some analysis to this effort that would advocate for conflicts if any occurs. In addition, we encourage to use the credit contribution sections for any paper that would highlight who did what. And for large teams, if any large paper come out with a lot of people involved, we might encourage alphabetical listing along with the credit contribution sections. Are there questions on this particular document, concerns, comments? Everything is good to raise at this point.
No policy on author order, is that right?
Right. So I think it's written in the document that the lead folks that are leading a publication would decide on the author's order. And if there are disagreement on the order among those with the other authors, the steering group will weigh in. Julien?
Yeah. I would like to know how is it possible to opt in for data contribution.
Data contribution. So like if you want to contribute data to the effort. Okay, so anybody can do that. There's a how to contribute page here that explain how you can help. And so if you want to contribute experimental data, I think the best is probably to go reach out to us and have a discussion how best to do that. Yeah.
Okay. All right.
If you have already collected data and you want to share, I think that you could also list it here, but you have to do the effort to explain how you think it's complementary and how to access and so on and so forth.
Okay. All right. Thank you.
Any other questions? And Richard?
Yes, hi, thanks. So just to clarify, with contributing code or analysis, so right now we, we have a human lab. We don't have animal research facilities, but we are quite heavily invested in computational stuff. So then we would contact a lead on a project, but additionally, we could also create an issue through GitHub directly, right? Is that
That's
what you
right.
have in mind?
In the analysis, in the project tracking page, there's a list of ongoing analysis projects. Most of this are placeholder at the moment. But what we anticipate happening is that when you start an analysis project, you should explain what is this, and if it fits within one of our analysis projects, just add your name. But so that there's visibility and everybody knows what's going on. I personally I like very much the radical transparency concept from Redalio, where you put yourself out there and explain what you're doing. I think that's really important for building trust. And so whenever I do something, I'm going to try to follow that principle of being very transparent. And I'm hoping that the rest of the group follow. Any other questions and comments? Okay. Yes, Karim?
Can you hear me? Yeah.
Yes.
So in case data has been collected under a slightly different variations of the stimulus that doesn't follow the 4 conditions.
Yes.
Who gets to decide, or what's the mechanism for deciding whether it's under the umbrella of the project or unrelated or marginally related?
I think I think this is a community effort, but I think if your project is listed on this page and you intend to be part of its collaborations, I think the individual labs have to do some effort to disseminate and share. If a labs wants to do something completely outside of this process, I think you can do so. I mean, it's fine with me.
What I meant is, that my lab is part of the contributing labs. But sometimes when one collects the first batch of data, we might want to rethink, for example, how we're doing the experiment, right?
Yeah.
Slight variations always arise. You want to collect it to try to get some trends better represented in the data. So it might not exactly follow, the experimental design per se. That's what I meant. So I think it's okay to share that and describe basically what is different from
that's fine.
The original project.
I think it would be beneficial if we discuss like publicly with the group, like I'm planning to do these changes. What do you think? And also I think leverage the expertise in the group as you discuss the design change. So for instance, I'm going to give you good examples. We had a couple conversations with Sarah Rudiger about the durations of the drifting gratings, and she highlighted that we really want to capture a full drifting grading cycle. And so the durations matters for some it's an hypothesis. Some of the labs might not care about that because you only care about the onset, but this is an example where like leveraging the group is helpful, I think.
Yeah, that's what I meant. Yeah, thank you.
Yeah, of course. Any other questions, comments? Okay. Hopefully my team sharing continues to work. Right, so we intend to add all of the meeting notes on this page here. So that's why I'm referring to it for today's agenda. Okay, so how do we track progress? So we made this project tracking page here, and it has several components. The first one is a link to a project board that is going to go on GitHub. In a minute, I'll introduce that. And we explain how to use this board. And there are several sub-projects that are listed. Some of these that are, I think, already started. So I'm going to mention them quickly. So the first is the stimulus design effort. And I've been working collaboratively with Sarah Rudiger already on this. So the intent of that project is to put together the first version of those 4 session types. And we keep this updated. So I think if you're planning to contribute data collection in your labs, I think it will be beneficial if you're part of this effort. And then I anticipate a number of future projects that will start once we have more data. One is data creation and sharing. So the intent here is how to package the data. So for OpenScope, we intend to use the Dandy and NWV system to share. And then there are analysis projects. For these, I essentially took the archive publications and extracted the project from the analysis plans. It might make sense to make more or break those down into sub-projects depending on what makes sense. Obviously, we don't have much data. We already do, but I think that probably is going to take off when there's a lot of more data set that are available. And so those are essentially what is encoded in mismatch response, the types of response more broadly in neurons, and then the comparison across different mismatch paradigm. And then there's also a project about computational modeling that I think will be very interesting to do down the line. But so I anticipate that those projects will change as we get started. But this is sort of a start of it. Any questions on this project list? Okay. I was planning to sort of go through this project board because I think it would be helpful for people to see how we are planning to track our progress at the Allen Institute. So typically an OpenScope project, the internal project, but the non-public ones, we take about a year to collect the data and share it broadly. And then the data is embargoed, is released on Dandy. So the teams that have worked with us at the end of a year, they have access to NWV files on Dandy. And then for a year, there's an embargo. And then the data is eventually released publicly on Dandy. This community project is meant to operate very differently. So we want to push data up as quickly as possible. And so it becomes important that you are aware of our progress towards this. And so to facilitate this, we decided to use GitHub tickets. And we made a public Kanban board with Anna and Ivana that is very visible for everyone to see. And so this means that we're going to create a subtask ticket that you can visualize with timelines. So if you go to some of these views, you can see that we have sort of started putting placeholders view. And so there are several ways to visualize this. You can visualize as a roadmap. You can visualize them as a board. So this is a traditional project management system called a Kanban board with items that have not started, things that are currently in the to-do. So you see that I'm tasked to test some of those stimuli in Bonsai that we put together on the eFIS and OFIS rigs. And then in progress, we are working on the authorship policy that you just saw, as well as the project management guideline that we were just discussing, and piloting some of the Bonsai work that is already started. So when we start data collection, you'll see like, oh, this has started there and it's finished there, and then you'll be able to sort of visualize our progress as it goes. I think it will be wonderful if external labs also use this system. I will be very happy to sort of give access to the board, and then people can create tickets. I know this is not the traditional academic practice, and so if one or several of the labs want to use the system, I think it will be great. But I will understand if you prefer not to. So that's how we're gonna track things at the Allen so that there's visibility. Are there any questions or comments on this? Okay. Let's go through this. Okay, so let's discuss a little bit about stimulus design and deployment. Okay, so this is a page I mentioned in project resources. We put together a number of sorry, I saw there's a question maybe. Oh no, it's not on this thread. A number of resources. So we have already started. So we at the Allen we're planning to use Bonsai for our stimulus design. And I'm hoping that Collaborating Lab can also do so. I think we recognize that Bonsai is different than what some of the labs are doing. So there are numerous systems for controlling experiments in the fields, BPOD, Psychophysics Toolbox, and many others. And so we had to choose one. The reason we chose Bonsai is that it's a complete open source stack. It has the capabilities to do a synchronous and asynchronous relationship in the data, which is quite powerful for experimental controls. And it's essentially like a visual programming language that has a fairly sized community around it. And so we want to help this ecosystem grow. So at the Allen, we sort of we hope to sort of use our resources to help grow component of the ecosystem. And so that's why we chose Bonsai in this particular case. So to help, we already started put together some resources. So the first one is Josh Siegel put together a really nice guide to understand what's different about Bonsai from traditional Python programming. And so I encourage you to read this because I think it really helps understand the difference in spirit of using Bonsai. And the second is a page called Bonsai Instructions. So the repository that you're looking at, this website, contains, so if you click on edit here, it brings you to the GitHub repo. And all of the script that we are developing actually in the code folder under stimulus control. So anybody can pull our scripts directly. And I'm personally working a lot with these, so I'm pushing a new version of the script regularly at this point. So let's go back to the website. So if you go to stimuli here, you see the list of scripts that we are working on. And so there are, remember that there are 4 scripts that we have to put together. The standard oddball protocol, the standard oddball with jitter, the sensory motor oddball protocol, and the sequence mismatch protocol. We already started working on 3 out of 4. So if you And for each one of these, I started to put together documentation pages. So if you go to the standard oddball protocol, there is a page that describes the content of the current script. Yesterday, I started to put together a figure. I mean, this is a work in progress, but highlight the part of the script. Sorry, I'm seeing there might be a question. Okay. And so you can actually access the current version of the script here in the script location. And then the page is meant to explain how the things is working. And it has a fair amount of details. And so the standard oddball protocol is almost fully implemented at this point. And so we were able to run this Bonsai script actually on the SLAP2 platform with Casper and Maede in the last few weeks. And actually collected data with it. So if you go to the experimental page here, there's a list of experiment that we have already conducted with the standard mismatch. And I want to point your attention to one of these that was done a few weeks ago, where you can access the script that was run here by clicking on this button. So it gives you the exact script that was run, as well as the experimental note from the experiment and access for the data on Dandy. So we have already collected glutamate release data with this standard mismatch, and this is a pilot. So we're sort of iterating and using this as a test for all of our system. And so and we converted this to NWB files that you can already look into. So
I just for the other experimentalists, before you go too far with this, I just want to say that the interfacing with the SLAP2 is basically no different from interfacing with most other two-photon microscopes in this respect. So you could probably use the stimulus scripts quite easily with with any, for example, scan image scope or probably other scopes as well.
That's right. So for me, it was really helpful to work with Casper because and Made, because we could quickly iterate the script, test it on the rig, and then have immediate feedback about alignments and so on and so forth. But now we are, in terms of script development, we are moving to implement those on the Mesoscope, the other imaging system, and the NeuroPixel rig at the Allen Institute. Okay, so the stimulus is here. Sarah Rudiger started putting together the standard oddball jitter. So I think she has a first version. I think it still need some work, but we have started. And I already started working on the sensory motor oddball. So in this one, we can already control. So the wheel under the mouse control the phase of the gratings was already implemented. So it flows backward as the mouse walks forward. And last week I started implementing the oddball in that paradigm. So we're sort of midway in this work. And the sequence mismatch protocol, I have not started working on that.
Question?
Yeah.
So hey, it's Andre, and we would be leading the primate studies as part of the collaboration. And in the primate, we don't yet have, in the lab at least, set up a walking protocol. But we had proposed in the archive paper to do it with a joystick and an avatar control. So I was wondering if I could get some feedback on that, as in terms of whether that would interface in a good way with the oddball that's being proposed here in mice, or if we should rethink that.
I think that could you could. I think there's probably several ways to modify the task. You could also do a joystick that control the gratings back and forth. I guess. I don't have fully formed ideas on this. Maybe what you want to do is, okay, so it's a good point. So let's go to this page. So you see, this is the current description of the stimulus for the mice experiments. And I put together a number of things. So the first thing is, if you click on the edit button on the top right, you can edit the markdown files and modify the content of the website. I also added a discussion link here. Hopefully that works. These start a thread on the GitHub forum linked to the repository where you can start discussion on a particular topic. And so, Andre, I think this might be the right place to have this conversation, where you could just start a thread and explain, I have this primate plan, I want to do that, and then we can discuss this on this forum. In addition, I think we should set up probably a bi-weekly meeting where we discuss stimulus design, and that might be the right place to also brainstorm in one-to-one. Does that make sense?
Yes, that makes sense. Thank you.
Alex?
Just very quick. So the Modo oddball had two oddballs. One was that the velocity gets discoupled, and the other one was that everything gets discoupled, and the optic flow is just completely random. Is that correct?
There's an open and a closed loop condition. Is that what you meant?
I thought there were two. So one was that they basically, the speed is coupled to their speed. And the other one is that it's entirely random what they see.
Yeah, so I think that would be, I think the intent was to pre-record a previous session where the speed of the mouse is varying and then replay.
So, okay, let me rephrase. There was one where even if the mouse stops, the flow would keep going.
I see. I don't think we had that in the document. If you think that's important, we could discuss it.
Okay, let's discuss it. That would be very important, yeah.
I want to add to what Alex said. During the discussions, I think there were ideas regarding adding gain modulation. So the speed would have either a higher gain or a lower gain that introduces the mismatch. So we can look at these variations without, without any complexity, at least at the beginning.
Yeah. In the bonsai workflow that I implemented, the gain is a parameter somewhere that can be changed very easily. So that's definitely possible.
Yeah.
I think I would like to test the forum so I know when we used the Google Doc, the comments were really handy to discuss these things. And my rationale to move for GitHub is because we have to track code and so much more that I think it makes more sense. And so if you could use the discussion forum to bring this up, I think that would be done publicly and everybody can see where we agree and disagree. And I'm going to do my best to follow the forum.
Yeah, and I suggest that before anyone edits the main page where you pointed that edit link, so that things can be tracked, we start the discussion topic here. And once there is an agreement, then the edit feature becomes, the next step. Otherwise, yes, people will just, insert their own modifications, and then no one is knowing what happened.
I could implement protections on the main branch of the Git repository if that's helpful, so that so far only there are very few people that have had a write access to the repo. I'm planning to add as many people that request it, but yeah. Okay. So I think we should, for the stimulus design, I think we should start this conversation on the forum, and then probably call a regular meeting to discuss this in the next months or so. I think every 2 weeks will make sense, probably, so that we align our development. Does that seems right to people? I think maybe this time is good for a weekly. Are there any concern if we do that? Okay. Good. The last thing I want to discuss, so we are almost at the hour, is analysis plan and tracking. Okay, so that's the analysis plan. So I want to sort of go back to we had 3 main hypotheses with a subset of analysis or describe in each one of these. And in the project tracking page, I use these to create a sub-project. And so I just anticipate that as the data is going to come in, we should coordinate analysis and have people say, I'm interested in analyzing this and this. To get us started on this, because we have a little data, I thought it would be helpful to start working on the SLAP2 data, because we already have, as I mentioned, we already have started an experiment. And we already have 1 assets on Dandy. We have started some analysis of it, but I want to do this with everyone. And I know Alex already started looking into it. But I think it might be a nice way to get ourselves into the mood of analyzing data. And so we have 2 experiments in SLAP2 that were collected. I must say, the team is extremely excited by this data assets. There are beautiful responses that are present and locked to the visual stimulus. So you're looking at glutamate release during oddball task in dendritic and apical dendrites. Like nobody has looked at that before. And so I would think that a number of people in this group might be interested in looking at this data asset. And so what I'm hoping we do is that in probably in 1 or 2 weeks, we have a discussion about the analysis of SLAP data with anybody that is interested join in. We can present analysis that we have done on this data. And then if anybody is interested in analyzing it, on the other hand, you're welcome to join and even contribute. Yes, Alex?
Can we have something like we have now for the stimuli, a discussion board, maybe for the analysis of the data as well?
That's right. So
for example,
I wonder if we could do a rotating. So like 1 week we discuss the stimulus and the next week analysis. Does that make sense?
I meant on the website.
Oh, like a discussion forum or?
Yeah, so right now for each stimulus, you have a page and you can discuss, which is great.
Yes.
So can we have something like that for the analysis? As you know, for example, I took your code that you guys wrote and I made it a collab, which some people might prefer. And so how can I share that, for example, so that everybody sees that?
Okay, I mean, there are several ways to do it. The first thing is you could create a page on analysis in the main website. So you can create a subpage. I can help you with that. The other thing is any experiment markdown here is also editable. And you can also go here and make a discussion on that experiment. And so, yeah, maybe that's the right way to start is here. And then as we do more analysis that are across multiple experiments, we can create a new page.
Yeah, just like you have for each stimulus type, we could have one for each analysis that gets started, maybe.
Yes, that would probably make sense. Any other comments?
Is it deliberate that the experiments are chunked by individual experiment and not by experiment type, for example, right? Because if we collect several dozen of these individual SLAP2 data sets, for example, we might want to group them together separately from other experiment types.
We could create subheadings, yeah. You're thinking on the website or?
Just in that left bar where you have experiments, that's going to get very crowded.
That's right. So let me show how it works. So if you go to the edit button, you go to the website. This website
we can edit this.
This is let me show. So this is a MKey Docs website. So it's using Markdown for content. If you click in Docs, there's a little where is it? Oh, it's upstream. Probably. Yeah, there's a little YAML file here called mkeydocs YAML. And this is essentially how the website is organized, the structure. So if you change here, you can create other pages. And anytime you commit to main, the website is updated.
Okay. Yeah, I will. I'll just read the. I just don't know the syntax in order to break it down. So it isn't deliberate that it's broken down by individual experiment. Great.
No,
That's
I mean it's
okay.
yeah, but we can organize this as we see fit.
Got it. Thank you.
I mean, I anticipate that as we collect more and more data, this website is going to get more and more crowded because we are documenting as it goes. But I thought it was important to already start setting a structure. The same way as when we started the Google Doc, we lay out the plan for the document. We have to lay out a plan for the experimental data collection. Otherwise, it's a mess. Shima has a question. Will there be a project board for analysis as well as track different analysis? Yes, we could do that. We could create tickets for analysis. I must say, I anticipate this community effort is going to have 3 phases. The first phases was the review. Now we're entering the data collection phase. And so it's very important that we document things very, very well. Once the data is finished, the analysis will likely last 2 years, and it's going to have its own, probably it's going to have its own practices and system in place that we'll have to figure out. But I think we can continue to use the website, but I anticipate that it's going to be a funnel. So like we review and then we collect everything under a shared umbrella, and then everybody was going to explore. And so I think it's going to be harder to maintain structure when analysis will start. That's my anticipation. Because people make discovery and then lots of small groups. Okay, any other questions? So I would like to propose that next week we have this meeting and discuss the stimulus design effort. And so I think it would be helpful if some of the labs that want to contribute experimental bandwidth spend time looking into the bonsai resources that are there. I know Sarah already did. And then the following week, we probably can talk about analysis. And I think it will make sense to discuss the analysis of SLAP2 data in more depth. And when we continue the cycle for now. Does that make sense? Are there questions and things that we should address before we part away?
I think that was a great rundown of a lot of details in the allotted time. So thank you, Jerome.
Yes, thank you, Karim. This talk was recorded. If you have any concern with us releasing this on YouTube, please let us know before we do so.